when I first heard about AIDS in the United States, I think it was in 1982. In France, we had very few cases at that time, but I read newspapers, some magazines, even scientific magazine about this new disease. But we were not very much concerned because there were only a few cases, and a few doctors in France actually were interested by this problem. There are very few patients.
And how did you get involved in AIDS research?
I got involved in AIDS research because I was working on retroviruses which can cause cancer, leukemia, and the idea that perhaps AIDS was caused by a variant of this kind of virus was on the air, probably came from the United States, but we heard it in France. And since we have already expertise on this kind of retroviruses, because we were looking actually for viruses involved in breast cancer. So it was not difficult for me and my team to shift a little bit to the AIDS problem. And this is what we did at the early January 1983.
I wanted to ask you about hemophiliacs. Because they had cell-free plasma, it was just the virus. But the virus sheds its membranes within 24 hours. So one thing we couldn't go is how was it able to actually infect the T cells?
Yes, it's a question, but we have to know that all the fraction of the blood could be infectious. And there is some virus bound to red blood cells which could be released also in the plasma after treatment or incubation. Some virus is bound to red cells also as well. So perhaps there are more virus. When you process the blood, more virus could come in the plasma, okay? And this virus could be protected by the plasma proteins from denaturation. This is one thing, one possibility. The other is the quality of the host. Hemophiliacs are fragile. Being transfused many times, their immune system also is depressed before they get infected with HIV. So they are prone to HIV infection because of their immune system weakness. Okay. So combining this with the fact that the virus could exist also in forms which are studying now, which probably are more resistant than we think for the usual particles, could explain.
The little ones.
Yes, little ones, or they bound to mycoplasma proteins. That's possible that mycoplasma are more resistant, perhaps. Mycoplasma envelope could be more resistant than the viral envelope.
Okay.
But there are all hypotheses. This is not based on solid data, of course. This is just they are just assumptions. But you are right. We have to explain why hemophiliacs have been so easily infected with plasma products.
In my interview with Dr. Flossie Wongstall and Bruce Walker, they both said that the mechanisms that HIV uses to kill T cells is still kind of unknown. Why is that? And do we have any new ideas of what might be killing the T cell or how?
Yes, there are in vitro many co-infections with bacteria, which we call mycoplasma, small bacteria which bind lymphocytes, also stay on the surface. And then when there is co-infection, the cell HIV and this one mycoplasma, they die very quickly. They form balloons. We can see that very easily in the tumor cell line, which we are using for growing the HIV virus. Okay. The question was, of course, this in vitro observation could be translated into in vivo situation. So we have spent a lot of time to see if people were co-infected also with mycoplasma. And we have some data encouraging with that, but we could not say one mycoplasma is always associated. Some mycoplasma are, but not one single species. There are several species involved. They could contribute to this also, to the death of the cells. But I would also involve another mechanism, which is oxidative stress. It's still more complicated. seems that in many infections, including HIV infection, you have a lot of free radicals produced, oxygen molecules, which are highly reactive and could damage DNA, proteins, lipids. So this is probably induced by some viral protein again, not the TAT protein, for instance, but perhaps also by other co-infections, mycoplasma, but other types of any types of co-infection. You know, many people in Africa have tuberculosis, they have malaria, they have parasitosis, and this activate too much the immune system and cause oxidative stress, and induce apoptosis. So the death of T4 lymphocyte, of course, is more important than the deaths of the other types of cells. But other types of cells also are prone to apoptosis. But most of the other cells are repaired by production in the bone marrow and with thymus. But probably also, since the virus infects the thymic cells, it may also have some induction of activation and then apoptosis, more specific on CD4 cells. It's complicated saying that we don't know, actually. We don't know which is the most important mechanism in this. And this is why I'm still working on that, and many other my colleagues are still working on that. It's one of the main basic problem of AIDS research, is to explain why the T4 cells die in many people, but not in all. Some people, probably because their immune system is strong enough to keep the virus lower, do not show this kind of cell death. But also I'm working on the fact that some viral forms are very small. I call them nanoforms. They are much smaller than the usual form, which is about 120 nanometers in size, in diameters. Here we are working with forms which are smaller than 20 nanometers, so about 5 to 10 times smaller than the normal forms. And it seems that those forms are more prominent in people which are treated by antiretrovirals. So this is probably part of the virus reservoir.
So those 15 nanometers, the viruses, that's HIV too? Or I'm sorry, did I misunderstand?
No, they are forms which are derived from HIV. They are HIV. But not the usual form with the membrane, you know, which you can see in your EM pictures, which have the size of about 120 nanometers. Here we are talking about forms which are less than 20 nanometers. So much smaller.
Are those infectious viruses too?
There are some infectious. They are not all infectious, but some of them are infectious, yes.
Oh, okay.
So this is something not published yet, but you can, you know, if your movie is produced, it's shown in a few months, it will be published. So.
Is that a new finding? Because Gelderboom told me that retroviruses are usually like one size, 100, 120.
Yes, this is the real size.
Oh, okay.
But, I want to explain why this virus persists, you know. And of course, there are several explanations, but one of them, it could hide itself in mycoplasma. It could also produce some very small forms which may not contain all the viral DNA or viral RNA, just one part. But those forms sometimes can be reconstituted into infectious forms. So my explanation of the fact that patients which are treated by antiretrovirals, they are fine, but once you stop the antiviral treatment, the virus comes back within a few weeks. My explanation, this virus comes from those forms, unusual forms.
Okay. And what is the ratio to the small viruses compared to the regular viruses?
I don't know, but there are many millions of those small particles,
okay,
which remains after antiretroviral treatment. Okay. If you look at the viral RNA, the normal size by molecular techniques, you find almost nothing. This is what we call virus load. It's decreasing almost to 0 or less than 550 copies per milliliter of blood. But here we are dealing with millions of those small particles which are not detected by usual PCR techniques.
Do they contain all the viral proteins as well?
No, probably not. No. Just a little bit of information, that's all. Probably a little piece of RNA. Oxidative stress occurs in any kind of infection, even chronic, mostly chronic infections. It can be also caused by bad nutrition if you don't have enough antioxidant in your nutriments. It can be also caused by chemical air pollution. So there are many factors which cause oxidative stress. Oxidative stress activate transcription factors which will make the cells divide or produce cytokines in a kind of immune cells, inflammatory cytokines. So you create conditions for cells to divide which are not exactly normal. So this first step of transformation. And the second step is that oxidative stress will also induce mutation in the DNA of those cells. So again, having an oncogenic effect by mutation.
How much does environmental toxins play in oxidative stress?
Well, air pollution, especially nutrition, could play a role. We don't get enough now of antioxidants from what we eat because of the globalization of agriculture. We also exposed to chemical pollution from the air, especially for respiratory disease, cancer, for instance, of the lung. And we have also some psychological effect on the immune system. Living in very large cities, we are more stressed, not oxidative stress, but psychological stressed than living in a small city or in campaign, in a rural area. So I think this also could play a role in decreasing our immune defense as well.
What would your message be to Africans who are suffering from potentially oxidative stress? Or not even Africans, but even in the West, how would you tell people to treat that?
I think at the end it will be done in a very rational way, in that you can be tested. You take a little sample of your blood, and you do some about 5 to 20 measures of the antioxidant, of the oxidative products, and so on. So you can have a profile of your situation. And according to this profile, you will prescribe products which will compensate for the deficit or the increase you have seen. Okay? And there are also some genetic factors. You have also genetic polymorphism in the antioxidative enzymes. So we have to do also that analysis. It's very complicated, but at the end, it will probably be applied to everybody in developed and developing countries. I think this is the key. Now, of course, we are not at this stage. What I'm just promoting is in a center we have been creating to have a laboratory for measuring oxidative stress and try some products which are accessible to African patients, like fruits extract and some herbal extracts.
It seems like everyone's just forgotten about poverty. And it's like the shift has just gone to AIDS now, and poverty's been pushed aside.
Well, AIDS is not the first problem, in Africa. They are used to die of something, die of malaria. Many young children die of malaria, still now. Many people die of cancer early. So it's just tuberculosis. Just AIDS comes on the top of this, and in the African mind, it's something, but not very different from the other problems.
What is your opinion on cofactors along with HIV?
Yes, I think I'm a promoter of the role of cofactors in AIDS, helping the virus to kill the cells like mycoplasma, but also reducing oxidative stress. And so any kind of, it has to be remembered that any kind of co-infection will activate the lymphocytes. And activation of lymphocytes is a key factor for the virus going well. The virus has to infect not resting T cells, which are our cells are mostly resting, but the few subsets which are activated are the target for the virus. So if there are more targets, of course, the virus will multiply better and sooner.
Are you still working on mycoplasma today?
Yes.
Quite a bit.
Well, I'm working on I've been working for several years in the physical association of mycoplasma with HIV. Because sometimes the mycoplasma, which is a small bacterium with a flexible membrane, can provide its membrane to the virus. Instead of the cell membrane, the virus will use the membrane of mycoplasma.
On the outer...
When it's binding to the cells, from the cells,
Okay.
because mycoplasma are also sticking very tightly to the cell membrane.
Okay.
So when the virus goes out, it could take some mycoplasma membrane or mycoplasma protein in this membrane.
Okay, I see.
So this is a way also for the virus to hide, because of course, if the virus is coated by a mycoplasma membrane, it will escape, of course, any kind of vaccine which will be directed to the normal membrane proteins.
So it shells the virus and protects it.
It could provide a shell to the virus. This is one way, but this is again laboratory observation. I don't know if it is really occurring in the in vivo situation, how much it has some importance, how much important is it in the in vivo situation, I don't know.
You talked about oxidative stress earlier. Is treating oxidative stress one of the best ways to deal with the African AIDS epidemic?
I think this is one way to approach to decrease the rate of transmission, because I believe HIV, we can be exposed to HIV many times without being chronically infected. Our immune system could get rid of the virus within a few weeks if you have a good immune system. And this is the problem also of African people. Their nutrition is not very equilibrated. They are in oxidative stress, even they are not infected with HIV. So their immune system doesn't work well already. So it's prone, it can, allow HIV to get in and persist. So there are many ways which are not the vaccine, the magic name, the vaccine. Many ways to decrease the transmission just by simple measures of nutrition, giving antioxidants, proper antioxidants, hygiene measures, fighting the other infections. So they are not spectacular, but they could, decrease very well the epidemic to the level they are in Occidental countries, Western countries.
If you have a good immune system, then your body can naturally get rid of HIV.
Yes.
Oh, interesting. Do you think we should have more of a push for antioxidants and things of that nature in Africa than antiretrovirals?
We should push for more, a combination of measures, antioxidant by nutrition, advice and nutrition, fighting other infections, malaria, tuberculosis, parasitosis. Most children have worms. Education, of course, genital hygiene for women and men also. Very simple measure, which not very expensive, but which could do a lot. And this is my worry about the many spectacular action. Of course, there are global funds to buy drugs and so on, Bill Gates and so on, for the vaccine. But those kind of measures are not very well funded, not funded at all, or they are, it really depends on the local government to take conscious of this, but local government, they take advice of the scientific advisors from the international institutions, and they don't get this kind of advice very often.
There's no money in nutrition, right? There's no profit.
There's no profit, yes. The water is important. Water is key.
Now, one thing you said, you were talking about the fact that if you have a built immune system, it is possible to get rid of HIV naturally. If you take a poor African who's been infected and you build up their immune system, is it possible for them to also naturally get rid of it?
I would think so.
That's an important.
It's important knowledge which is completely neglected. People always think of drugs and vaccine. Americans are very. They think, they have the Pasteurian scheme in mind: one disease, one agent, one treatment, which holds true for the acute infections. But when you deal with an acute, even with a chronic disease, it's quite different. And you have to involve several factors, including cofactors, to explain everything. Otherwise, if he was right, if you were right, AIDS should be, we could get AIDS within a few weeks after a HIV infection. This is actually the case of a few primates. Some monkey die of some strain of SIV could induce a very short disease. But the natural history of AIDS in humans is different, and you have to explain why.
Why do you think it is one cause, one effect, and that's it?
Well, this is, I think, a philosophy of science, positivism. It's positivism behind probably this. I would say also is also very well spread in Europe. It's Cartesian logics. Cartesian logics. Compartments, but I think we see now some limitation of this, in medicine. If, for instance, for polyarthritis, rheumatoid polyarthritis, you treat with anti-TNF antibodies. Of course, you stop the inflammation, but you get cancer. Some people get cancer after that because you are acting on something which has a positive role in the inflammation. It's a reaction of the organism to something. Is good by the definition. And you don't think you have you have to treat the cause behind this. You are just treating the symptom. And most of the medicine of the West actually only treat the symptoms, which is a mistake. We have to treat the cause. And people accept to have cancer, Alzheimer, comme ça. God, bad luck. No, they are precise causes. We have to find out.
Do you ever get to work with John Moore at all?
What?
In New York? Do you ever get to collaborate with him?
No, not really. Not really. No.
No. Oh. Don't like it?
No. I have nothing against him. No. It's also the one which deny completely, I think, cofactors. He doesn't like the idea, probably. Isolating a virus is something you don't see first, of course. You don't see the virus. You see the virus only later with electron microscope. But what you see is some activity of the virus. And we were lucky by the fact that in 1970, a specific enzyme of this kind of virus was discovered, called reverse transcriptase. It's an enzyme which can reverse transcribe the DNA into RNA. Probably some cells have it too, but at that time, only some viruses have this enzyme. So if you find this enzyme activity in a supernatant of a culture of cells, you could say this comes from a retrovirus. Okay. So this is what we did first, actually. Of course, in order to for a retrovirus to be produced, it has to be produced by cells. So the first step to isolate a virus is first to cultivate some cells. And we were fortunate in that since AIDS was a disease of lymphocytes or white blood cells, we knew how to cultivate this kind of cells. Not from our own experiments, but from current knowledge at that time. We knew how to grow T cells, and Dr. Gallo will tell you that he isolated in his laboratories, some of his colleagues isolated a T cell growth factor, which was very key to keep T cells growing in culture. So we use actually this kind of factor before working on AIDS to cultivate T lymphocyte from women having breast cancer, because we thought the virus which could cause breast cancer was also growing in T lymphocytes. starting from a mouse model. So in 1982, I actually asked Robert Gallo to send me some of T-cell growth factor just to keep T-cell growing to isolate this breast cancer-associated retrovirus. But in 1983, of course, my early stock was gone. So I asked one of my colleagues at the Saint Louis Hospital in Paris to bring me some of this T-cell growth factor. And thanks to this, we could isolate HIV.
Is that interleukin-2?
Yes.
Okay.
It was called later interleukin-2, but at that time it was a crude material, just supernatant of some activated T cells. So we had in ants, we have the factors to grow the cells, and also I have interferon, because we showed before in my lab that the growth of retroviruses in cells is highly dependent of the secretion of interferon. If there is too much interferon, which is of course secreted by cells which are infected by viruses, they will stop the virus multiplication. So in order to improve the virus isolation, I use serum which will neutralize human interferon. So I use anti-interferon serum. I think it was also a key factor. Of course, later we found that it was not always necessary to isolate some HIV strains, but I think at that time it was important.
Okay.
So we have a T-cell growth factor from Gallo. We have the anti-serum against interferon from my colleague in France, Ariane Gresser. And so I had also the skill to dissociate cells from lymph nodes. Lymph nodes are very tight glands, and there is a technique which needs to be used to dissociate the cell without killing the cells. Okay? So I have some kind of homogenizer called Downs homogenizer to dissociate the cells of the lymph node I got from the patients, from the doctor who I took the biopsy. So I have this is only technical. I don't see any invention in that. There is no real discovery. But the fact we have in ants in the same person, three things: anti-interferon, T-cell growth factor, of course some skill also to cultivate cell, because I've been cultivating cell for decades before, made actually was a key factor for the early success, because the first experiment was the good one. It was right. We could isolate this activity, and this is thanks to my colleagues and collaborator Françoise Barré-Sinoussi, who had learned the technique to detect reverse transcriptase activity. So we have all the techniques taken from the others, but we could, gather them to in one piece, and it worked.
What are the characteristics of HIV?
Well, this is, I would say, a usual retrovirus. If we think of this very specific subfamily called lentiviruses, retrolentiviruses, is very similar. We cannot distinguish HIV from simian HIV or from a Vicenna virus or from a goat retrovirus. It's the same. So the morphology is the same. What I would say that HIV-1, there are two viruses in man, HIV-1 and HIV-2. HIV-1 loses its knobs at the surface very easily. HIV-2 is a more typical morphology in which, like HIV, the simian retrovirus, it keeps most of its knobs on the surface.
Would I be right in saying that from the pictures alone, though, in culture, you can't prove what virus you found yet?
Of course, electron microscopy is not sufficient to prove you have a retrovirus. That's clear. You need other characteristics like the density. The reverse transcriptase activity is the key enzyme, which is at that time was only associated with retroviruses. That's how we describe it first. We didn't describe the electron microscopy as a proof. We described the reverse transcriptase activity as a proof it was a retrovirus. But here also we were criticized Some people from Gallo's lab saying, are you sure you have a reverse transcriptase activity there? Could it be a cellular DNA polymerase? We had this kind of question.
To silence them, how come you guys didn't just show pictures from the gradient instead of just the culture?
We first show it from the culture. Just by centrifugation, but not sucrose gradient, just by making a pellet of the virus, you could look at it also this way. But here, of course, you have many impurities, coming from the cell. The sucrose gradient has the advantage to partly purify the virus, but again, even in the band of the virus, you have also cellular vesicles, which have the same density, but not the same look, of course, at the electron microscope. The characteristic of the virus is this dense core, the dense conical core, which is typical of what we call retrolentiviruses. The same in sheep, the same in horses, the same in goat, the same in men. Well, I would say again, nobody has completely isolated the virus. Well, it depends on what you mean by isolation. Isolation means you can grow up the virus in cell culture. We did that. And we could pass it continuously. We had a permanent system, to keep the virus going. It was not the same culture, but we could pass one from one culture to another. Okay. So I think it was virus isolation. I argue about this. I will argue. But this is past, we have so many discussions in the past now. It's over.
Can you talk about the purification process and what that entails?
Of the virus?
Yes.
Yes, of course. When you have this reverse transcriptase activity, you have many other things in the medium, in the culture medium, and how to purify this. Of course, the amount of virus is very small at that stage, and there are techniques of purification by differential centrifugation you can use. But when you have a very so small amount of virus, I don't think we could see anything. So the first stage was to amplify the virus production. And the first culture died off within weeks, 4 weeks. They were lymphocytes from the Brou patient. So at that time, I have to pass the virus into a second culture. And I did not know, of course, if all kind of lymphocytes from blood donors would work. Fortunately, it worked. We have the small blood transfusion center at the Pasteur Institute, so we could have access to lymphocytes from blood donors. And it was, I think, a Spanish donor who eventually was there to give his blood. And we succeeded in producing the virus again in the second culture and so on. But the amount of virus was still very small in terms of reverse transcriptase activity. So we looked for other types of cells, especially tumor cell lines, so without having access to blood donation, having some some T cell derived from blood, but going continuously in culture, from leukemia, for instance. And we tried many at that stage. None of them worked. Unfortunately, because we used this virus from the pre-AIDS patients. If we had used the virus from the full-blown AIDS patient, we would have succeeded. But at the beginning, we did not know that. So we had problems to grow up the virus in large amounts, and after that, we sent this virus to Dr. Gallo's lab, which also had no success. But a second time, he had success before us from our own virus. Why? Because without knowing it, we had a third isolate from a full-blown AIDS patient called LAI, first 3 letters of his name. And this patient had a virus growing very quickly, very strongly, not only in lymphocyte, killing them, but also on tumor cells, tumor cell line. But we didn't try, actually, on the tumor cell line we have tried before, first for the first Brou isolate. We didn't try again on this isolate. And without knowing it, there was some mixture. The virus from the third patient could overgrow the virus of the first patient. How it was mixed, we don't know, because we are skilled biologists. My technician also was very good. So I wonder why in our own lab there was some mixing. But the fact is, it was some mixing. So the third isolate overgrow the first isolate. And when I sent knowing it was, thinking it was the first isolate, actually I sent also the third isolate to Dr. Gallo's lab, who had the tumor cell line at work, and he could grow his colleague, his collaborator Popovic could grow the virus before us. We also finally also were able to grow the virus, not the same cell first, but what we call a B cell line transformed by EBV, but this is technical, in October 1983. And just a few months later, thanks to also to the collaboration with Robin Weiss, which was in London, we also could grow the virus in a very good T cell line in April 1984. So there are details, but it's difficult to explain to the lay people what happened. But it's clear that there was some exchange. It's normal to have some exchange of viruses. But I still wonder why in my lab, in Gallo's lab, in a few other labs, this new strain called ADI grew over the other strains.
Once you were able to get a lot of the virus, can you then explain that the purifying process?
The first thing we tried, of course, was to see the virus. The electron microscopy. I have a good electron microscopy in my lab. So I asked him to make section of lymphocytes from the Brou patient. And he showed me a few pictures of virus, which, very rare actually. And the trigger in my mind was a few months later when I could produce this LAI virus in more quantities and partially purified in sucrose gradient, because retrovirus have a very specific density. And in a sucrose gradient, you have a range of density starting from 1 to 1.3, for instance. And the virus forms, if you centrifuge those particles, the virus forms a band, very sharp bands at 116 density. This is the density of retroviruses. And Françoise Barré could see the activity, the reverse transcription associated at this peak of 116. It was not a complete purification, I must say, because again we didn't have enough, and only when there was some mass production, not by ourselves, but also by the company which was associated with the Pasteur Institute, which made several liters. They could have enough particles to see, of course, homogenates of viruses at the electron microscope. Okay. This is something we have been people which criticize the HIV/AIDS, HIV being the cause of AIDS, have raised many times. You don't produce much of the virus, but unlike some other retroviruses causing leukemia in chicken, for instance, you don't see much of the viral particles. They are there, of course, but they have a strong effect, even distance. They have a strong effect, even there are very few. So we have sensitive techniques, biochemical techniques to detect this virus by reverse transcriptase, by antibody, of course, or by antibody binding. But in the body, there is not much virus circulating. Very few particles are found in the blood. At one time, a given time, there are probably more particles in the lymph nodes and some other tissues, but it's not a very large infection in terms of viral particles. So the mass production can be obtained by from tumor cell lines, but again, mass production doesn't mean a lot of viral particles.
Once you cultured the virus in the flask, you then spun it. Now, what was the evidence that you found that showed you there was a virus in the material? The
evidence was given by the biochemical activities, these enzymatic activities called reverse transcriptase, which was done by Françoise Barré-Sinoussi. She learned the technique actually in an American lab, in an NIH, not Gallo's lab, but another lab in the 70s. So it was at the time a classical technique to detect in a very sensitive way retroviruses. The biochemical activity, reverse transcriptase. It means you have to take a piece of synthetic RNA. You give it some labeled radioactive precursors, and you make a DNA strand, and you detect the DNA strand.
And then what was the next thing you did? Was the next thing to take a picture, or what's the next step?
No, the next thing was to keep the virus growing, and also to try to characterize the proteins of this virus by antibodies specific to viral proteins. And we use first the antibody against HTLV1 proteins because this was the only known human retrovirus at that time, HTLV1. So I asked Robert Gallo to send me the reagents. He did. And we found that those reagents did not react with the protein of our virus. Okay? So there was an important clue. And after that, of course, we have to develop techniques, ELISA techniques mostly, to do some epidemiological study. How far this virus was spread in the gay community, in people having AIDS or pre-AIDS. This was, of course, a very important argument to show that a correlation between this infection and a disease. Took some months for that.
Did you get the correlation before the science papers, Gallo science papers?
No, the first 1983 science paper, our paper, or the paper by Gallo, just say we had isolated a virus, and we were already convinced that this virus was different from HTLV1 at that time. Gallo was not. But after that, we did some work with our colleagues from hospital, from the university, to really do some epidemiological study with the virus we had in hands, which was not large amounts. We tried to grow it in a mass. First we failed, we succeed only later. So I would say by the end of 1983, beginning of 1984, we had a good feeling our virus was the cause of AIDS, but not many people were convinced we were right. And the confirmation came when Gallo published had his own results. So I would say, this is, I think, admitted by both of us, we were the first to isolate so-called HIV. But for proving this virus was the cause of AIDS, I think we have both an important role. I think Gallo has an important role. We had some roles as well. That's what I could say.
When purifying, it's important to photograph the density gradient where virus is banned. Why is that a crucial step?
Well, Gederblom, I know him well. He's a good electron microscopist in Berlin. And actually, he gave me the best picture of my virus.
Oh, really?
Yes. Well, of course, in order to purify, you have to make this sucrose gradient density equilibrium to have a sharp band. And if you take that sharp band, you have almost pure. Not completely pure, because there are cell vesicles which have the same density. This is why you don't see in the picture, you have a mixture of cellular vesicles, and viral particles.
So, is it really important, or is it not really important?
For us, it's not important. But some people say, if you don't have complete purification, how do you know the disease is not caused by something else? It's very unlikely, I would say, because now we have very specific inhibitors of the viral enzymes. And those inhibitors not only inhibit the virus, they inhibit the disease itself. Okay. So this is a good a posteriori proof that HIV causes AIDS.
Now, one thing When you purify HIV, there are some challenges because it's contaminated with cellular debris
As I said,
and particles that look like retroviruses but are not infected.
yes, as I said.
How do you distinguish between what is infective and what isn't?
You cannot. You cannot. It's admitted in the retrovirus world that most of the viral particles you see are not infectious. Only 1 in 1,000 is infectious. But you cannot say, you cannot discriminate. You have just to know that the titer, the infectious titer, is much lower than the physical titer, number of particles.
Does the EM help you distinguish, though, what is what?
No.
Because there's contamination, it's difficult to figure out which proteins are actually associated with the virus so that you can create a specific and accurate test. How do you get around that?
Well, first we did not actually. It's true that we have a very good staff, and so there are many contaminants. We did a technique which is called RIPA, which means radioactive immune reaction, in which you label the proteins by radioactive precursors. Then you treat with the antibody of the person which is infected. You compare with the antibody with the blood of someone, serum of someone who is not infected. And you run a gel of the proteins, you do autoradiography, and you see the bands, the bell bands, which are recognized by the antibody of the person which is infected. Okay? So in this case, you can see the difference, and from the difference, you can say this is virus specific, this is not. This is what we did. So we had identified 2 proteins first, the P2425, the core protein of the virus, and another protein called P18. And it was only after many months we could distinguish also the surface glycoprotein, GP120. Gallo used a different technique, the Western blot technique, which also could detect specific bands, comparing serum antibodies from people which are infected to people which are not infected. We have to do always a control, of course, in this case.
Do you still feel P41 is actin?
Yes, we did.
Oh no, do you still think that today?
I don't know, of course not.
Okay.
But at that time, yes, we had a discussion with Gallo and his collaborators, because he found the GP41 as identified as a viral protein, and we had in our technique protein having the same molecular weight, which was in fact actin. We were both right, or both wrong actually, because it's true that the virus can bring with him some actin protein, even inside the viral particles. Even you purify, you can still have actin. But it's true that there is also a protein coded by the viral gene called GP41, which is a protein which is below the surface protein, called the transmembrane protein.
I see. So how come you guys only were able to identify 2 of the proteins?
Yes, well, the one key factor was the sequencing of the viral genome, the cloning the DNA, and looking at what we call open reading frame. We can see, knowing the genetic code, the translation from 3 bases in 1 amino acid. We could say the open reading frame, which could be translated into proteins. Then we could identify 1, 2, 3, 4, 5, etc. Taking into account what we knew already from animal retroviruses, we could assign to each protein one function. But the problem with HIV is too many proteins. Some are structural proteins, the envelope, the core protein, but some are just so-called regulatory proteins or accessory proteins, which are made during the replication of the virus, which are very important, but are not part of the viral particles. They are made inside the cell. So it took some time, but the team were so anxious to be the first that there was a race. Identifying in Gallo's lab or Hazeltine lab, the TAT proteins. Flassig Monstall probably told you about the TAT. We identified 9. sorry. We were we were very proud to identify this NEF protein, which was missed by the American group, completely missed. And then we decide to actually the first those proteins were just called by letter. So after that, we have a discussion on the phone with Gallows and other groups, American groups, to give specific names. So the TAT name came, the NEF, the VIF, and so on. We were very proud to to give to baptize our new proteins.
Did virus production play any role in that too? Was there a problem getting a lot of virus in the gradient?
No, because we have to look for those proteins inside the cells, not in the gradient, not in the supernatant.
Oh, you don't.
So the key was to identify the open reading frame on the DNA sequence, and then from the DNA sequence we can deduce the protein sequence, and also try to see by antibody. Natural antibody found in the patients to immune precipitate those proteins having the same molecular weight. So we could deduce the molecular weight of those proteins and try to see them in gels. But the sucrose gradient purification didn't play any role at all in this identification of the small protein. It played a role for the for the core proteins of the viral particles, but not for the regulatory or the accessory proteins, which are made inside the cells. That was found later to circulate also around outside the cell, but at the beginning it was found inside the cell.
Okay, I think I was taught wrong then. I was taught that the purification part was to get all the proteins from the virus.
No.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus,
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus,
Oh, just to make sure it's to make sure what you thought you had is what you had.
You have a virus which has a property. We have a particle which has a property of a virus having a sharp density band. A cellular vesicle could have several densities, but the viral particles having been made of all of the same proteins have always the same density, with some variability depending on the cell which are producing the viral particles, because also there are membrane components coming from the cells on the viral membrane.
So when you got to look at the EM for the first time of the purification, what did you see?
But you see an increase of typical viral particles, which correspond to what literature has shown already for other lentivirus from animals. Yes, so actually we find also viral protein shedding very early. Actually, the problem of HIV proteins first, it was easy to characterize the core proteins, but not the surface proteins. It took many months before we could see, and Gallo thought he had the surface protein, basically it was the transmembrane protein called GP41, but the large surface protein called GP120, nobody could see it first because the viral strain we were working with could shed this protein very easily in tissue culture. We could hardly see them in the electron microscope, and also by antibody testing. So it took us several months, and also not Gallo, by another American, Max Essex or Myron Essex, you probably heard about this name, also found this glycoprotein. I think it was at the end of 1984, or the fall of 1984, and we also could find it. But we could see in sucrose gradient, most of this protein was staying away from the viral particles because they were detached from the viral particles. So for some unknown reason, the virus can shed very easily these surface proteins.
Does that have any implications in HIV pathogenesis?
But perhaps, yes, because this circulating surface protein could have some effects, like I told you before, some effects on uninfected cell. It could bind the CD4. It could have induced apoptosis in uninfected cells.
What was going on in your minds when you first started doing the experiments back in 1982, 83?
Well, at that time, we were not very much excited because we thought it may be HTLV, the virus described already by Robert Gallo for leukemia. So we look for reagents trying to characterize this virus, HTLV1 reagents, and we asked Gallo, which kindly sent us the antibody, specific antibody against HTLV1. And the excitation started when we saw that those reagents did not react with our virus. So it seemed that it was a very different virus. And the second excitation came when we could see it at the electron microscope, because this virus has very different morphology from the HTLV-like virus, from the HTLV-like virus. It was very close to a subfamily of retrovirus called lentiviruses, causing chronic diseases in animals, in horses, sheep, goats, not AIDS, of course, but causing long chronic wasting disease or encephalitis, pneumonia, and so on. So at that time, which was June 1983, I really was excited because we knew it was a new type of virus, not shown before in man, and very likely to be the cause of AIDS. Of course, at that time, we didn't have the full proof it was the cause of AIDS. It was associated with the disease, but since AIDS is an immune-depressing disease, you could have many passenger agents, viruses, fungi, anything which could be associated but be the resulting of the immune depression, not causing the disease, not causing the immune depression, resulting of it. The proof came a little bit later when we could associate the serology with the disease. HIV positive, at that time it was called LAV positive, positive patient had AIDS or pre-AIDS. So there was a study done by my colleagues, Françoise Brun-Vézinet, which was forgotten in the film the band played on, who conducted this part and showing that indeed most of the patients with AIDS had antibody against HIV. And a new proof came when we could isolate the same virus from African patients. At the beginning, we worked with the viruses isolated from gay men, infants, or hemophilic children. But in September 1983, I got some virus from African patients which were hospitalized in France, and it was the same virus, and we had the same type of antibody. A study made a few months later, in February 1984, with Peter Piot, Jonathan Mann in Zaire, in Congo, also showed that patients with clinical AIDS had antibody against our virus. Almost 100%.
And that was the proof of the causation.
It was a strong indication. But at that time, we were only a few to believe in that, like a small group, about 10 people. And there was still, the idea that HTLV1, variant of HTLV1 could cause the virus, but everything changed in April 1984 when Robert came to my office and showed all his data, published also in Science, that really HIV was the cause of AIDS, having more isolates than we had. But we had, I would say, in April 1984, we had about 5-6 isolates from several types of patients, including African patients.
Do you know who Professor Kurth is?
Professor Curtis?
Kurth at the Robert Koch Institute?
Robert Koch, of course, yes.
He said the electron micrograph he published in his mind wasn't proof for the existence of a new retrovirus. He said it could be an arenavirus, which also buds and has spikes. He was not convinced. How would you respond to critics like that?
Actually, I didn't heard about Kurt criticism, but from a French electron microscopic, her name was Françoise Haguenau. We had a meeting in France in, I think it was in the spring of 1984. And she said the same thing, that it was not a retrovirus, it was another. Because, people had in mind that the leukemic type of retroviruses, most of the retrovirus studies at that time were oncogenic retroviruses in animals or HTLV in men. And this virus type of virus are very typical morphology, which HIV had not. But the key was to compare the viral particle we could see with the lentivirus of animals, which are also retroviruses, not causing cancer, but causing long-lasting diseases, Visna, the disease called Visna in sheep, which was described in Iceland. The anemia, equine infectious anemia virus from horses. There was no discussion comparing the picture we had and the picture we could see in the literature of those animal retroviruses. But electron microscopy were not familiar with this kind of virus. They were familiar with the leukemic type of retroviruses. But I completely discard this criticism, and I laughed at that at that time, because it was really nothing,
Some people are very fortunate that they don't have these side effects.
What is it?